Welcome everybody to the Image Processing and Image Analysis workshop. My name is Andreas Grieser and I will be your host for this session. This workshop will be split up into 3 parts of each 30 minutes. Between the parts we will have breaks of 10 minutes in which you can ask questions. You can also ask questions in the chat during the sessions. Please ask them in the open chat for everyone so everyone can profit from the answers given. During the last few days, everybody of you should have received an email containing a download link to a Geodict 2021 version. You should also have received a license for Geodict 2021 that should be valid until after the user meeting week. We will use this Geodict version and this license for this workshop. Additionally, you should have received an email containing a download link with the data required for this workshop. The zip file you've downloaded contains the input data required to do this workshop and also the results that we will be producing. So if at any point along the way you get lost due to some technical issues, there will be points where you can rejoin the process. So a short introduction of myself. I'm a business manager for image processing and image analysis at Mastermarket. But I'm also a software engineer and doing my PhD on the topic of image analysis, especially for fibrous media. I've studied computer science at the University of Kaiserslautern, and I've been with Mastermarket since 2014. In this workshop, we will tackle the topic of importing images into Geodict and processing them, and also running some analysis on the segmented images. So the goal at the end is that you are able to handle image data in Geodict, know how to import it, how to apply image filters and create segmentations of these images, and afterwards run analysis on the segmented images, for example, to obtain grain size distributions or fiber diameter distributions. In this workshop, we will use 2 different datasets. The first one is an NMC cathode out of a battery, taken from the Battery Microstructure Library of the National Renewable Energy Laboratory of the United States of America. That scan was done by the University College London's Chemical Engineering Department. And the second one is an air filter media, as you can purchase it for a car, where we ordered a scan on ourselves. So now let's get started with Geodict. As this is the first workshop of the week, and importing images is naturally also the start for working with Geodict, I will start with a short introduction on how to set up Geodict and how to use it. So I've here installed my Geodict, and I will launch it by double-clicking. Now Geodict is starting. And I get the message that I don't have a license installed, which is probably the case for most of you. So if I press OK, I get into the Geodict license dialog, and I can install a license by selecting Install License File and clicking on Find License. Now Geodict is in the default project folder, and I can choose my license file that I have stored in the folder with the data for the workshop. It's a .glig file, which is probably new for the people that have used Geodict before, as we had to change the file ending to avoid some collision with other files. So now I open it. And it takes a bit of time because it is a floating license that is run on the cloud and sometimes takes a bit of time to connect. So, but it connected, it's all fine, and the license is set as default. I have to accept the license agreement. And then I have to wait for a bit. Successfully installed the license. Now again have to wait a bit because this is a floating license and there are options to not lock all the modules that are part of this license with each Geodict run. This allows, for example, if multiple people at a company are using GeoDict, but one is only, for example, using FiberGeo, while the other one is only using GrainGeo. They do not log each other out of the license. So, but for our case now, we just log all the modules as it is by default. I click OK here. And yeah, it already tells me the license will be valid until the 4th of October. So now I'm on the GeoDict Mason screen. Let's make it big here. And now you can see the main GUI of GeoDict. You have up here the bar with icons for quick access, for example, to load structures, to load result files, to create videos or pictures or save structures that you have in GeoDict. And on the left, you have the current status of GeoDict. So you have the CPU usage of your computer, the RAM usage of your computer, and the disk usage on the current disk where the project folder that is listed down here is set to. Below this status, you have the modules GUI that changes depending on what module you select. Currently, we are in FiberGeo, and most of these module GUIs look the same. They have up here a combo box to select a function, and below it an options button, an edit button to change parameters of the function you want to run. So for example, if I click here on edit, I can change the parameters that I used to generate a fiber structure. To actually run the process, we then have to click on generate below here. And also very helpful button is the help button next to it, as it will guide you directly to the user guide of the corresponding module. On the bottom, we also have now collapsed the console where GeoDict is generating output. So in here, we right now see the messages that we logged all the floaters for the different modules, that the license was installed successfully and stuff like this. So there's often useful information given here. On top of the console, we have the recent files and the recent project folders. Right now, it's an empty list because I have cleared all the settings. And maybe most importantly, up here, we have the different menus, which are called import, model, analyze, predict, and export, where you can select the actual modules. So right now we are still in FiberGeo, but as we want to import files, we want to go into import and select import GeoVol to import volume data. Before we start importing our image data, we will switch our project folder to the folder containing the data you have downloaded. So we click right on the project folder right here, click on select project folder, and switch into the folder that contains the data for the workshop. Now the project folder has changed. You can also change the project folder up here in the files menu. To start importing data, we have to click on the browse button in import GeoVol. Next to it, you can see a list of all the different file formats GeoDict supports, but we will just leave it at all files and click on browse. And then in the folder named cathode NMC 398 NM, we find a lot of TIFF images. So to import them, we select the first one, click on open, and then we get into the import settings dialog. And in here, we make sure that import all files is checked, and we have to enter the correct voxel length. So we switch the unit to nanometers and enter the value that was given in the folder name of 398 nanometers. Down here, we can now see it will import 625 slices and require 2.1 gigabytes of memory. To start importing the images, we click on Image Processing. And we get a progress bar where GeoDict is importing all the files. And then the image processing user interface will open. Before we go into more detail on the image processing, I will go back to the main user interface because we haven't seen the actual interface yet when some data is loaded. So right now I'm in the 2D view of the loaded data. You see there is a new collection of tabs up here where you have a lot of view settings. Right now the image is just black because I'm at the first slice and there is nothing in there. But if I pull on this slider, I will slide through the dataset now in the X direction, and we can see a 2D visualization of the dataset. To zoom in on this, you can click and hold the right mouse button, then you will be able to zoom to the point where your mouse is. And to move the image around, you can click and hold the left mouse button and move it around. If you want to jump to defined zoom stages, you can click on this button right here. It will jump in even steps to get you zoomed into example to twice the size or 4 times the size of the original image. On the right side, you can see the color bar corresponding to the image, and you can see it's a 16-bit image as it ranged from 0 to 65,000. But it's also very dark, so we are probably not using the full range of the image. In the bottom, you can see a scale bar. You can switch the direction in which you look at the image by clicking here on direction and, for example, clicking on Z. That is the view that we've seen in the TIFF images as they are in the folder because they are imported in Z direction. If you want to change the color bar, you can go here into the volume field tab and click on the edit button for the color bar. So, for example, I can open this up and switch the color bar range here a bit down. And then we'll see the contrast between the grains and the pore space a bit better. Of course, we can also look onto the image in 3D by clicking on 3D up here. That takes a bit of time because the Raycaster has to build its acceleration structure. But it should soon be done. Here we are in the 3D view. Right now we again don't see very much interesting stuff because the outer areas are all solid, but we can use the clipping sliders here in the volume field tab to clip into the field and get a view like this. Also maybe clip away from the top of the Z direction to see a bit better into the 3D structure. And also we can of course enable transparency by clicking on this button. That again will take a moment. Then we get a transparent view and we can see a bit better the grains here. I hope this transmits well through the WebEx connection. But if not, it's not that important right now. We can just disable transparency again because it makes the computer a bit slow, as you can see on the load graph here on the left. Again, takes a moment, so now we are back in the non-transparent view. That's a lot faster. What might be an interesting feature is if you pull on the clip slider here, you can basically emulate a simple segmentation of the image with a certain threshold. So we can actually get an idea of what our final structure after image processing might look like. The controls in here are, by the way, similar to the 2D controls. You have to zoom by pressing and holding the right mouse button, and you can turn by pressing and holding the left mouse button. You can also zoom by using the mouse wheel. To pan the structure around, you can click and hold the mouse wheel like this. Now let's go back into the image processing interface that we ignored earlier on. So let's click down here and open the second window and directly make it big. So this is the image processing user interface. You have the three main views in the X, Y, and Z direction here in the middle. On the top you have some settings, and on the right bottom you have a histogram of the gray values, a 3D preview, the gray value distribution in X, Y, and Z direction. And on the left here you have all the options you have for image processing. Now, first of all, this is a bit small, so by pressing and holding the right mouse button, I will zoom in here to get it a bit bigger, and also I will move around a bit these bars to get a better view of the actual XY plane. You might be wondering why your image is rendered that red now. That's because we have a segmentation overlay enabled up here. But that's not really interesting to us right now. We just reduce the visibility or just turn it down to 0. In general, over here, you can control overlays. There are different overlays, for example, for cropping the image, for rotating the image, for shear correction of FibSim scans, for ring artifact removal, for example. I will show that one. And if you can see here now, there are these rings drawn on the image, and you can move them around. So if you have ring artifacts in your CT scan, you can put this overlay right aligned to the ring artifacts and then apply the filter. More of this later. And yeah, now we can just go back to the normal segmentation overlay. Yeah. Also on top here, you can control the gray value range that is visualized. As I have said before, this is a 16-bit CT scan. And as you can see, only the gray value range from 0 to 12,000 is visualized. This explains why there's such a nice contrast compared to what we saw before in the 2D and 3D view. And it's also visible down here on the right in the histogram, where the maximum value is near 12,000. You can change this. You will see that the contrast gets worse. And we have some values here in the histogram that were basically before pulled to the maximum brightness in the image. But as this is a logarithmic histogram, these are really not a lot of voxels compared to the major part of the CT scan. So we pull this back to, we put the value back to 12,000. And we can see we can also actually pull up the value from 0, because the outer area is where the 0 is happening, but we don't actually need 0. We can increase this a bit. So now we are right where the slope of the histogram starts, and we can see now brightness of the grains, and the contrast is a lot better. Now let's go back into the actual image processing tools. Here on the left, you can see the tree view that contains all the tools you need for image processing. There are two tabs. The first is for image processing, and the second one is for segmentation and labeling. We will for now focus on the image processing tab. And first of all, we want to remove all this empty area around here and crop out a nice squared piece in the middle where everything is full of the actual material we want to analyze. To do so, we open up the image size subtree and go into the crop menu. Now, one thing that happens is that automatically our overlay switches into the crop mode. Now, it's not really that visible now, but you see that there is a tiny yellow line up here, and you can grab that line and move it down. You can also just enter the values directly here or pull on the sliders, and you will see the area that will be cropped from the image. Now we move this here and here, and also down a bit here and move it up here. To get to the data we actually want to analyze, we will crop it to something like 250 and 750, and also in the y direction 250. Let's take 200 and 700. Get to something 500 by 500. And also in the Z direction, we see there is a lot of empty area. So we want to crop that too. And to do so, we put in here and in here. Something like this. We can scroll down to the view to see if we catch everything. So there is no empty area more in here. It's all solid. Yeah. And now we can apply this crop. Maybe make it a nicer number. Drop in a bit more to get to 260 and click on apply. Now Geodik crops down the image. You can see it's a lot smaller now. It's only 500 by 500 by 260. Normally that's not an issue to have a bigger image, but for the sake of having fast computations in the workshop, we will only do such a small sample now. So after cropping the image, we can now have again a look on the histogram down here. We can see, okay, there isn't the peak at the 0 more because we excluded all the outer space that had a 0 value. We again can change the gray value range to better fit our actual value range. We can have this 3D preview here where you can see the image. And also what this might be interesting now is to inspect the actual gray value distribution in the different directions. So here we have the mean gray value for all the X slices plotted, and also the fit through the mean value to see if there's any trend. Because often you get in a CT scan the issue that you have like a gradient in your image, and that you would be able to see here. So for this example, we only have a change of less than 100 over the whole thing for a gray value range of 2000 to 12,000, so that should be fine. The same does apply for the Y gray value range, and also for the Z gray value range. Everything should be fine. You can see that there is some noise happening here, but that is due to the size of the grains being relatively small and just creating this change in mean value. If they would not be fine, you could go in here in the image processing tree in the gray value brightness adjustment tab, and for example do a gradient brightness correction if there would be a gradient in here, but we don't need this now. So one thing that we now see, and what is very obvious in the CT scan, is that there is a lot of noise. So if I zoom in here, see there's really a lot of noise, and we want to get rid of this noise. So we go into the collapse these other trees, and we go into the image filter subtree, and in here we can see all the image filters we have available to us. There is, for example, the non-local mean filter, which is the pretty much go-to filter to get rid of noise without blurring your image too much. But there are also other filters like a Gauss filter or a sharpening filter. All these filters are GPU accelerated and pretty fast if a good GPU is available, especially for the non-local means filter. That's usually more on the computational heavy side. This is very helpful. So we will be able to use this even for a short demonstration in the workshop. On top of this usually known filter, there is also the option to apply, for example, a user-defined filter, where you can just write a Python script in which you access the data from Geodict, apply some filter. Maybe if you've wrote one yourself, you can use it, and then you will be able to integrate it directly in here and even enter some parameters using the Geodict Python GeoPython variables that are explained in the other workshop right now. On top of that, we also have specialized FIBSEM filter for alignment and thickness adjustment, and very interesting is also the curtaining filter that is able to remove the streak artifacts you often see in FIBSEM images very well without blurring your image. The same applies for specialized CT filters. There we have a filter to remove the ring artifacts that I've shown you before. Now, as we already approaching the 30 minutes mark and doing the filtering and segmentation now would be a bit tight, I decide we will now save the image in the state we have it, so we can later load it and then do the break. So to save the image, we click on File and then click on Save as 3D image. Then we can select the format we want to save the image in. So GRW is a Geodict format. It's called Geodict RAW because it's basically a header, and attached to the header is the RAW image. But we also have added the option to compress it. So I would always go to this format if you want to use the file with Geodict again. But you can also again write TIFF, for example, or just a RAW file. We will go with GRW now. We select the compress option and then we save as 3D. By default, this save dialog will open in our project folder that we've set earlier on. And now we need only to define a file name. So we name this thing cathode NMC cropped. There's no need to enter the voxel length in the file name anymore because in the header of the file it is saved automatically and Geodict will read it again when we load the file. And so we save the file. Now Geodict writes the file. It takes a moment. And now we have it saved to our disk. I will for then now say we do the break. And after the break, we will continue with filtering this image and segmenting it and doing some analysis on it. You can now ask questions if you want to.